Great. Okay. Well, it's so great to talk with Dr. Michael Pelosi. This is a real privilege for us. Dr. Pelosi has been an HIV cure researcher. He wants to cure HIV, not just suppress it, for many years. And he's been digging deep into HIV reservoirs, and which could come in very handy with long COVID, I assume. But more to our purposes, he's one of the founders of the LIINC project, the how is this long-term impact of infection with novel coronavirus project at UCSF. And that project has just mushroomed. It's kind of like the project we always wanted in MECFS, you know. And so thus far, LIINC got going very quickly with the pandemic. It's enrolled over 1,000 participants. It's gathered over 20,000 biospecimens, and it's associated with over 60 researchers and staff.
Yeah.
So I guess my first question is, did you? It seems like you started gathering biospecimens early. So is that correct?
Yeah. So yeah, just a minor correction. My last name is pronounced Peluso. I've
Luso.
no
Okay.
relation to the congressperson.
Oh,
Um,
Pelosi.
uh, but yeah, yeah, I'm Michael Peluso. I'm at UCSF, and like you said, I came here almost 8 years ago now, really to focus on understanding, why HIV is an incurable infection and how we could develop different virus targeting therapies or immune system targeting therapies to try to cure people of HIV, either to eradicate it from their bodies or to um, hone their immune systems to be able to control it without medications. And that was my main focus until March of 2020, and I mean it continues to be a focus. But what happened in March of 2020 was that, and I remember it very clearly. I was sitting actually in my HIV primary care clinic, uh, and we got basically a notification that a person in the emergency department had been diagnosed with COVID, which was a thing that was, happening, but it was exclusively pretty much happening in people who traveled. And the unique thing about this notification is that this person had not left the city in
Mm,
years.
okay.
And I remember just getting this feeling like, oh my goodness, this is everywhere. And that's what this means. This has to be everywhere. Um, what are we going to do? And at that time, there was such intense focus, as there should have been, on understanding, who would get COVID, who would get sick, who would end up in the ICU on a ventilator, who would, die. Um, and we had a meeting of our group, me, Steve Deeks, who's my mentor, Tim Henrich, who's another mentor, others, to think about, what role could we play in trying to address this problem. And we realized that, everybody was so focused on the acute COVID question, and nobody was really focusing at that time on what happened to people when they got to the other side. And we thought that that was going to be important in sort of informing our understanding of the virus and, potentially treatments or preventative methods. And we basically decided to invest our energy in that. And so, um, you know, uh, everything kind of was shutting down at that time. And um, we assembled this team of people who were quite, influential, way before my time in the days of understanding HIV. to try to apply that same framework to understanding SARS-CoV-2. And basically, on Zoom from our living rooms, we constructed what became LIINC, and it was, really remarkable, um, just how um, how everybody just came together to do this. Sort of everybody sort of dropped what they were doing. We would meet 6 days a week on Zoom for hours, into the night, to plan this. And this included everybody, included the faculty, included an army of medical students whose medical school was thrown into total disarray because they couldn't be there anymore. Um, Staff members, study managers, research coordinators, laboratory people, regulatory people. And we constructed this, and we didn't know about long COVID at the time. We constructed this to bank specimens, which is actually what you asked me, to collect specimens and store them, and to figure out what we would do with them later. Um, and we, got IRB approval remarkably quickly. The fastest turnaround I've ever had. Great support from UCSF. It was amazing. And we opened our doors, in mid-April of 2020 to start seeing people who had had COVID a few weeks prior. Um, and basically, within a couple of weeks of opening, we began to get outreach from people who were saying things like, I really want to be in this research study, but something's not right. I am just still, I have this unbelievable fatigue, or, I've been off of work for 6 weeks now because I'm, cognitively not where I was, and I'm a healthcare worker and I'm afraid of going back to work. Or, even one participant who was like, I need to do the visit lying flat because I've not been able to be upright in a
Yeah.
sustained manner for 6 weeks. And um, we were hearing all of this, and this was also around the time um,
Was
where
that shocking to you that people
I,
were getting so sick?
um, you know, I was aware of people having sort of chronic manifestations of acute infections. I didn't have a lot of experience in terms of ME/CFS, which would have been the most useful experience to have had. But like with EBV, Epstein-Barr virus, or other infections, I had seen this, and I had actually had some personal experience with it, you know, a decade or more prior, and so I believed what I was hearing, and I had read in the there was an article in the New York Times in early May about COVID survivors in Italy. Because if you remember back,
Oh yeah.
the initial wave in China, and then the second wave was really in Italy, even before the East Coast of the United States. And it was about how these Italian survivors of COVID were experiencing prolonged symptoms, and so that was right around the same time. And I basically, I said, this is what we are seeing. We need to be systematically capturing this. And so, because we were meeting 6 days a week in the evenings, I sort of brought this to our epidemiology experts, and we revised our study instruments to basically make sure we were systematically capturing these, prolonged the official symptoms of COVID at the time, which were on the CDC website. There were 10 symptoms or something, but then some of this other stuff that we were starting to hear about. And we did not specifically focus on people who had those symptoms, but we wanted to make sure that if people were having, what became known as long COVID, that we weren't missing it. And so we were seeing, vastly more people who were fine, had, gotten COVID and fully recovered. But once or twice a week, we would see someone with long COVID, and so we sort of through that became one of the earliest long COVID studies that was specifically focused on also biospecimen collection. That's how it began.
Yeah. Did you
Um,
think it would get this big? Are you surprised?
I am surprised. I guess a few things. So I when we initially designed LIINC, the duration of follow-up was going to be 4 months. Um, and so, there was June of 2020, a couple of things happened. So there were reports of this from other places too, other places in the US. Um, Hopkins had an early study of long COVID. They were seeing similar things. Study in Australia was seeing similar things. Um, and UCSF, to its great credit, actually like convened a grand rounds on this topic in June of 2020, which I think may have been the first academic grand rounds on the topic. Um, uh, there was also sort of a receding of COVID around that time. And we sort of, that was the first time where people wondered whether we were out of the woods. And so we were a little like on the fence about where things would be going after June of 2020. But unfortunately for all of us, COVID picked up again. And we basically said, this is probably going to be a long, uh, the pandemic is going to continue. This population of people who have long-term symptoms are going to continue. We need to make this a longer study. And so we basically recalibrated the study to accommodate that in July of 2020. And that summer we extended follow-up basically to 2 years. Again, too short. And said, well, we can't possibly see people every month, so we'll see people every month for the first few months, and then we'll see them every 3 or 4 months, for 2 years. And so we reconfigured everything to do that. We realized that we were going to need to, further sort of professionalize our biospecimen repository, um, and and then, it has been continuous since then. Since then we've extended follow-up to indefinite for people who want to be followed indefinitely. Um, and the shift of the program has really been toward understanding the biology of long COVID. Although, of course, we have questions separate from long COVID. That's really been the main focus, since the end of 2020. And then, if you're interested, we can talk about sort of moving from studying the breadth of this to sort of the depth of it,
Yeah.
which has been, a focus of the last few years.
Has it, you noted in a paper recently, I think that we're in our fifth year of COVID and long COVID. It seems like it just started yesterday to me, in some ways. Um, but is there anything that surprised you about how things have turned out?
Um, I mean, I don't think in January of 2020 I would have predicted that we would be here having this conversation. I think the things that have been surprising, they're I'm sort of a pessimist by nature. uh, Although I try not to be. And, I think one thing that was surprising was, there were sort of multiple declarations of victory throughout these 5 years on COVID,
Mm,
right? So there was what I said in June of 2020. Then in June of 2021, before Delta, I mean, things were looking really good in May of 2021. There were very few new cases. Re, you know, it looked like the vaccine was going to get us basically eradication. There was a lot of hope, and I remember that feeling in the end of June of 2021 when institutions were thinking of kind of that it was going to be over. I said, well, we sort of thought this last year, like maybe let's see what happens after July 4th. And then Delta came. And then Delta sort of waned and we got into the fall of 2021 and it's like, well, the holidays are coming up. And then Omicron. So, the roller coaster has been a little surprising. Um, I think, the other thing that was surprising, you know, not being an expert in infection associated chronic disease when this started, um, having expertise in chronic infections, but not being an expert in infection related chronic disease. Um, I think, the context in which long COVID emerged was just so unique and, you know, I hope we will never have that again. But we probably will never have that again where, you know, the whole population is immunologically naive to a virus and it emerges basically simultaneously everywhere and everyone is at risk. Everybody knows about it. Everybody is, there were issues with testing, sure, but like everybody was testing actively, were wondering whether they had this. And then people, because of the efforts of the patient community to raise awareness of what became known as long COVID, people were sort of broadly aware of the post-acute condition. And so um, it's sort of surprising that we were set up to be able to study something like this with rigor, really for the first time, because most other infection associated conditions, for the most part, sort of emerge sporadically, even when there are concentrated outbreaks of ME or it often it's all sort of understood retroactively. And so that I don't think anybody could have predicted that we would be in a situation where it may actually be possible to figure this out. That was surprising. And then um, I think a good surprise, although I'm not sure it's a surprise, is um, I think one of the pleasures of this has been the level of collaboration, sort of scientifically, and also with people with the condition, which is something that's has always been true of at least my generation's work in HIV. Um, but I think that has been really one of the main sort of joys and privileges and benefits and motivators, is the people we study or participants in our research study, but getting to know them and be a part of their journey with this terrible condition, is very special to me, and I think that's true of many people working in this field. We feel like we're kind of in the trenches together, and I think that's really cool. um, yeah.
Good. I hear that a lot from MECFS researchers. Once they get really involved, they're really, and long COVID, they're really fascinated by the disease
Yeah.
and struck by what some people go through. Uh,
Yeah.
but is there anything in particular that's really exciting you about the field right now? I know I read, a couple years ago, you said there's no simple answer. There's no simple answer to this, And then I
Yeah.
think, with the antiviral trials, that's not a simple answer either. Uh, so it's very complex,
Yeah.
It seems like there's a lot of different avenues, a lot of different, you know, approaches, a lot of different things people are studying. Is there anything in particular that's really exciting you right now about where the field is going?
Yeah, I mean, I think what's most exciting to me, is, a relatively young person working in this area, is just how sustained it's been and how great the collaborative science is and how much of a sort of feels like we're all on the same team, and that's very special. That's not a given
Hmm.
in science and academics and life. Um, I think that that's that gets me going every day. I think
Well,
that
there's more of a sense of collaboration that,
there's a huge collaborative spirit, a huge
yeah.
team spirit, a huge and exciting feeling that we're making progress. I mean, to me, whenever I see another group's work, it is exciting to me because we have hypotheses that we are testing and we are finding stuff, and sometimes the stuff that we find surprises us.
Hmm. Hmm.
Um, and okay, progress is slow, whatever. That's true. But in the scheme of science, it's going at a pace.
Hmm.
And that's exciting. It feels like every day when I open my email, I will see something new. That's exciting. There are these great newsletters that I get every week that I learn from, and just the pace of what is happening and the number of people working on this is it's really neat to be sort of a small piece of that. Um, the other thing that I don't think people see is that, one of the real challenges in this field was that it was sort of a group of people that assembled with a common interest in understanding this that never actually met each other for so long, right? So many of us were started working on this in 2020, and we were just faces on the Zoom panel, Um, and it wasn't until um, you know, probably even, I guess it was 2023 that people started being able to meet each other in person at these, scientific meetings once they started to slowly reemerge in ways that tried to be safe. And that was really galvanizing. And a lot of those meetings, you know, are closed meetings or meetings that people don't see. Um,
Hmm.
but when we have those meetings, it is so energizing because everybody just talks about their ideas, talks about where they think things need to go. Um, it's really exciting. And so what I'm excited for this year is, there's going to be the first meeting that really did that was this meeting put on by the Keystone Symposia group
Hmm.
a couple years ago.
Yeah.
And that was the first time many of us were in the same room together. That really galvanized people. And there's going to be a sort of a follow-up meeting this year
Yes.
in the summer. I'm really excited for that. The other thing that's exciting is that um, we're starting to, even if results of these trials or the first round of trials or whatever is negative, it's exciting to see that clinical trials have been done and are reporting out. And it's exciting to see the level of rigor with which most of these studies have been conducted.
Mhm.
Um, that's really encouraging to me. And, you know, there was a real possibility that, the first few of these trials would report out and would have negative results, and then people would sort of throw up their hands and walk away. But that's
Right.
not at all been what's happened, right? What has instead happened is that I really believe that the first round of negative trials has opened the door to a much broader swath of future clinical trials that are informed by what happened in the first round, and that can explore a broader set of biological mechanisms than just the virus. So that's neat to be observing and to be a part of. Yeah.
Oh, well, I watched the Recover treatment symposium or meeting, and it seems like they're ready to take on you. I think you suggested, things like platform trials and combination trials and small trials that really attempt to dig into the pathophysiology, which you're doing with the Arrow trial, I guess Arrow 2 or whatever it was. And I guess some people did improve in that trial, and you're kind of trying to figure out what happened with them.
Yeah.
Uh,
Yeah. So
uh,
yeah.
so do you see more of that with Recover and other areas?
Yeah. I mean, the I don't know a better way to say it, but the vibe of the the meeting that you're referring to last year, the initial convening of Recover TLC, sort of the plan to start planning was very positive.
Yes, yes.
Um, and that I was a a great first step, and I'm really excited to see what that sort of network will decide to pursue. That being said, not everything can or should be done within Recover and Recover TLC. And what's been encouraging to me is that there are lots of sort of parallel infrastructures that all inform one another that are being set up kind of around that. And
Hmm,
so,
hmm,
the NIH has been really, great about uh, funding research within Recover, but also separate and complementary to Recover.
hmm.
I have, funding separate from Recover to look at different biological mechanisms within LIINC. And so that, the sort of overall infrastructure supporting this work and driving it forward is like really important. Uh, we participate in Recover clinical trials, which are amazing because they can be done at scale. Having sites all around the country and, um, you know, having a network like that is just so valuable. We've seen how valuable that is in other conditions like in HIV with the AIDS Clinical Trials Group, the ACTG, really important in driving progress. But we know that work that happens outside of the networks, like we do, in LIINC and like we do in our HIV program, is also really important in informing what the networks should be pursuing, right?
Hmm,
Can't
hmm.
do everything, can't, there are not infinite resources. And so, our group um, is really, we contribute to these larger efficacy studies, but what we love doing, our MO, our passion is these smaller proof of concept studies, um, where we identify the biology that we want to target, whether that's viral persistence, whether that's immune dysfunction, whether that's, herpes virus reactivation, EBV reactivation, and, autoimmunity, whatever. And then we design these really intense, and they're not for everyone, um, pathogenesis studies to probe that biology. Um, and, you know, we sometimes get criticism. People say, oh, the study's small. Small studies can still see a big effect. And what we're looking for are big effects. Where they say,
They're kind of small but deep.
yeah, we're small
Yeah,
but deep.
yeah.
Other criticisms are, it's really burdensome to participate. That's 100% true. It is. You can't do it remotely. Also true. But we have seen in our work in HIV how important it is to have these types of studies as
Hmm.
as a sort of a complement to the larger, lesser burden, um, less intense, more generalizable, easier to participate in studies.
Right.
And like as an example of that, you know, uh, before I was working on COVID, actually simultaneously to the pandemic emerging, we were running this HIV cure trial. That was this, and if you'll let me talk about HIV for a minute, it was basically this gauntlet of immunotherapy. It was a single arm study, so no placebo arm, and people were in this study getting immunotherapy interventions for 34 weeks. These experimental vaccines, monoclonal antibodies in combination, toll-like receptor agonists to alter immune function. And then
All at once. They were like in
in
one person.
series, yeah, in one
Yes,
person,
yes. Really, really amazing stuff.
yeah, and then at the end of it, we stop people's HIV. We ask people to stop their HIV meds, which we never ask people to do in clinical practice, right? And we watch what happened. And in this study, and we have a preprint from the study. It's under review now. It had 10 people. And normally, if you took 10 people and you stopped their meds, all their virus would just come back immediately, right? What happened in the study is seven of those 10 people, seven of them went on to have totally atypical ability to control their HIV in a sustained manner for months, in some cases over a year. And that study is not going to cure HIV, right? But that study has opened the door to so much more in terms of understanding why people succeed or fail at, their immune system being able to control the virus. And we think it really moved the needle. And that's a 10-person I mean, I saw those people more than I saw my family during COVID, right? Super intense study. And that's the framework that we're taking for this. We
Okay.
need, accessible, large trials that people can do from home, who can't make it into a medical center, or don't have a medical center near them. But we need both. It's not all or none. And so, um, anyway, I hope that anecdote is maybe convinced some people that there's value to these intense studies that really just try to move the needle, so to
Yeah.
speak. And that is sort of the bread and butter for our group. But that's not what people think about often when they think about clinical trials. So yeah,
Right. Right. That is a super intense study. And what would you need to be able to get to the place where you could say, we want to give 10 long COVID patients an immunomodulator, an antiviral, whatever, these different potential treatments. Would you have to know a lot more about long COVID? Would you have to, you talked about, uh, I think you said something that was really interesting. You said, we cannot ignore this anymore. We don't know which types of patients are in our studies.
yeah,
Uh, so really winnowing out the subsets, and I guess you're working on the Viper program now. Is that going to be key to just be able to molecularly differentiate the different subsets? And
yeah,
I just have another question. I mean, what about these subsets? I know COVID can affect the kidneys, it can affect the liver, it can affect the heart. Da, 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 da. I would have thought by now that we would have like a long COVID liver subset and a long COVID kidney subset and a long COVID lung subset, but we don't have these yet. And I wonder why. And I also wonder if, as we've seen in MUCFS, we see these these kind of eruptions in the beginning, and then things seem to kind of tend towards this fatiguing post-exertional malaise-like condition. And I wonder maybe
yeah,
if that's what's, over time, what happens in
yeah. No,
all
it's a
long
great
COVID
point.
patients.
Yeah.
Sorry, I threw about 5 questions in there.
Yeah, I mean, I think that the goal of the first round of clinical trials was appropriately to be as inclusive as possible because we needed to kind of use um, those studies to understand, A, whether we would just hit a home run, which would have been
Right.
great, but the likelihood of that was low and didn't happen. Um, and two, whether we could learn anything about how to better design the next round of trials. And um, you know, we are still waiting on the more in-depth analyses from all of these studies, from Stop PASC, from PAX-LC, from Outsmart LC, our monoclonal study, um, to really kind of get those answers. But I think um, it's clear that one of the potential explanations for why um, the top line result of all these studies was negative is that we have too much um, variability in who we're enrolling. And that's not to say that I, want to be restrictive, but we know in science and epidemiology, and it's a statistical issue, right? Where um, if you're not focused enough in who is being recruited into a certain study, it actually affects statistically your ability to observe an effect if an effect is really there. It's a statistical mathematical issue. Um, so I would not, it's easy to critique, it's easy to criticize, it's easy to say how you would change things in retrospect. But honestly, I wouldn't change anything about the first round of studies. I think we needed to do them that way, and I think that that's fine. We've learned stuff, and we can now design the next round. You mentioned Viper, so I think I hopefully I won't get in trouble for saying this, but I think that there was a real um, concern about and sort of resistance toward emphasizing too much, you know, the biomarker type measurements as we were trying to launch the first round of studies, because I think people were rightfully concerned that we didn't know what was meaningful in terms of measurements. And the result would be that we excluded lots and lots and lots of people. Um, a consequence of that, I think, was that we underdeveloped the pipeline development, the pipelines for biomarker development. We didn't invest enough in them. And as a consequence of that, now we're in a situation where we don't really know how to select people for the next round of trials, aside from what is happening now, which is that, certain studies are focusing on specific phenotypes, symptomatic groupings, rather than all comers. So we developed and we got some pilot funding last year from the Convergence Institute uh, to begin piloting this program called Viper, which is basically focused on trying to solve the biomarker problem. Because the way that biomarkers are being developed for long COVID right now is that every lab that wants to develop a biomarker is doing so based on their own cohort, which we can't really understand, and on samples that are collected for this purpose, but can't really then be provided for validation in another lab or for head-to-head comparison against another biomarker. And actually, the biggest problem in all of this is that in order to develop a good biomarker for long COVID, we can't really be making a comparison between people with long COVID and people who've never had COVID. That doesn't make a whole lot of sense because everybody's had COVID. Even if you think you haven't had COVID, you've almost certainly had COVID. And so the comparison
Yes,
needs to be between people who have long COVID and people who had COVID and are fine, have fully recovered.
right.
And that group is woefully underrepresented in every biomarker study that's been done so far.
Oh,
This recovered group. There are these papers about biomarkers that have, claimed to validate a biomarker based on 10 fully recovered people. I mean, that's just that's never going to hold up. So anyway, the purpose of this program that we call Viper is to basically turbocharge biomarker development, not just for the initial mechanism that we were interested in, which is viral persistence, but actually across all different mechanisms of long COVID, with a view toward informing who to enroll in these clinical trials. And so, that program, again, we've learned from HIV. There's this really important program in HIV called RAVEN, also a cool acronym, um, that was all focused on biomarker development in HIV and understanding different measures of the HIV reservoir. And so Viper, the spirit of Viper, is kind of based on the success of that program called Raven, and basically trying to apply that to long COVID. And we're hoping that we will get sustained support for this to expand beyond the pilot, to a much larger program, which is what we'll eventually need, all with the goal to informing how to get people into clinical trials.
And the pilot is focused on viral persistence.
The
Showing that the virus, being able to show that the virus is there in blood or saliva. Uh,
the pilot is focused on assay validation related to viral persistence, but
mm,
the key to the program is that we're not just collecting samples for one assay, right? The key to the program is actually the cohort. And so the idea is that we are able to, even more rigorously than we do in LIINC, intensely sample people in a concentrated period of time who have
mm,
really deeply phenotyped long COVID or not. And then this is not a, single tube blood collection. This is a lots of samples, multiple different sites, that all get cryopreserved to develop assays that we have and can think about now, not we, but that exist and we can wrap our heads around now. But also, if someone comes up with a great assay a year from now, we can then apply that assay to these samples and say, how does assay, you know, assay like N or P or S, like, down the line function compared to assay A, which is the where we're at now. So this is not just for now, but it's also sort of thinking about the long-term plan here, being able to make valid comparisons even as we move forward in time. So that's sort of the vision for this. We'll see if we can get it across the finish line.
well, you guys are great at long-term planning. That's one thing that's clear. Really building a foundation for the field and being able to move things forward. I remember Deek said that the surrogate biomarker for HIV, which was I guess it was RNA,
Yeah,
was just so effective in being able to develop
yeah,
treatments for it. It was like a turning point.
yeah,
Um,
it was a total game changer. Yeah.
Difficult with the coronavirus and long COVID, being able to tell if the virus is persisting or not. Is it
Well, it's hard to make a direct comparison because, in the early 1990s, a test that we totally take for granted, PCR, was only just developed, right? And so in the early days of HIV, I mean, I was in kindergarten in the early 90s, but so I was not there to see this firsthand. But um, you know, in the early days of the HIV epidemic, it was a clinical, AIDS was a clinical diagnosis. It's a syndrome, right? The S in AIDS is syndrome. And it was defined based on ultimately the presence of opportunistic infections, and then
right?
eventually, people's CD4 T cell levels. But it took, you know, a decade of HIV research to get to the point that we can measure virus in blood because the technology just didn't exist. Um, we've tried to apply those technologies to SARS-CoV-2, and it has not been straightforward, as you said. But that doesn't mean that there's not a technology that's being developed right now that will ultimately be the answer to this. So it's harder. It is true that it's harder to apply the same exact technologies that caused the breakthrough for HIV, but we have to remember that those technologies were only developed at that time. So,
Right.
um, but yeah, do I think that every single person with long COVID has this driven by viral persistence, by SARS-CoV-2 persistence? I think probably no, but I don't know. Um, uh, so do I think it's going to be the answer for everyone? Probably not. Uh, did I believe in this 5 years ago? No. Do I believe in it now? Yes. Um, I've, gained more historical knowledge that maybe I should have believed in it back then. But ultimately, like seeing this with my own eyes and our own cohort has made me a believer, that this is driving disease in at least a subset of people. The problem is we don't know how to easily identify those people, and the most straightforward treatments, um, don't seem to work for everyone. So, uh, I think there's so much nuance here, and what you said, about setting things up for the long term, I think struck me because this is a long, this is a long game, right? Like if this was going to be easy to solve, we would have solved it in 2020. That doesn't mean that we should give up.
Hmm. Hmm.
It means people who aren't in this for for the long game should probably not be doing this. But what's, getting back to your initial question, what's so inspiring and amazing to me about the people who are working on this is how invested they are. There were a lot of people who were in this field very transiently and, made contributions or not. And but the people who are working on this now, I think really are in it, with a view toward the long game. And that extends, that also extends beyond COVID and long COVID. I think we're all now thinking about how we can apply what we have learned or are learning or will learn about long COVID to other conditions like ME/CFS and to conditions that many people probably aren't thinking about, like post-Ebola syndrome, which is a thing and
Right.
remarkable overlap. And we are building out a program on that. Even though it doesn't affect um, uh, you know, it's a sort of a different type of epidemic. Uh, we're thinking about post-Lyme syndromes and chronic Lyme, different type of pathogen. And we're thinking about the future because, if there is a situation where another pandemic virus or other pathogen emerges, we don't want to have to create this all from scratch again. And
Right.
I fully understand that people who were working in MECFS before COVID emerged feel like this shouldn't have had to be created from scratch for long COVID, which is absolutely true. But there was too little investment in MECFS before then, right? So, but now I think we've seen how this can happen at scale, and even though no one wants another pandemic to happen in our lifetimes, and I hope and pray that it does not. If it does, we need to be ready to go. And so we're also thinking about, how this infrastructure that we've developed um, for long COVID can be applied to non-COVID MECFS, but also can be applied in the future should something else emerge. And so we want to be ready for that.
Yeah. Yeah. That's been our big hope in the MECFS community. We were selfishly encouraged when COVID-19 struck and long COVID.
Yeah, I don't think
And
I don't think it's
yeah,
selfish. I mean,
yeah,
it's it's M the MECFS work has so many additional challenges because there was, often wasn't this unique confluence of
right,
events. And even when there was, in the 80s in Incline Village or similar, a lot of the technology just didn't exist at that time, right? And so, I worked on uh, a commentary with Maureen Hansen, um, which was really fun, and I learned so much from that. But we talked in that piece about how many of the technologies that were rapidly applied for long COVID were just sort of a dream in the 80s and 90s, and so um, the context is really really unique, and I don't think that can be sort of overstated. Yeah.
right, right. Does the fact that long COVID is such a complex disorder that, it's if it's a viral persistence, it's hard to determine that. And then there are all these other similarities, so many similarities between these different post-viral conditions. Does that suggest that they're all kind of being created in similar ways, even though different pathogens are triggering them?
Yeah, I mean that is a reasonable theory. There could be differences upstream and then sort of convergence downstream, and um, I think that that's why it's become so obvious that it's really important to study these conditions alongside one another, not to necessarily lump them all together, but uh, the cross condition comparison is going to be really important in answering that question. I think that we're seeing desire or directive from groups that are funding this type of work to have that, to support studies that are trying to do
Great.
that. I think that's good. I don't think it should be a requirement, but I think it's something that we're thinking a lot about, and there are lots of sort of added benefit to doing that for sure. Um,
Okay.
yeah, I think, the other thing I would say is, yeah, it's complicated, but um, I think one of the reasons why I love infectious disease within medicine, right? There's sort of a spectrum of comfort with uncertainty, right? There are some medical specialties that exist in a realm of like great certainty. um uh and there are others um that are just more comfortable with uncertainty. I remember when I was in training, having a heated discussion with a vascular surgeon, um, who was basically saying, I just want to know, is there an infection or not, right? In this person's aorta or some, like, some inaccessible space. And I remember, reflecting on that and the sort of desire for certainty, which is what we all want, and um, recognizing that fields like ID are often much more comfortable with uncertainty. And so I think that that's okay. I think it's okay if there is, people try to undermine this field by saying, oh, there's too much uncertainty, or there's too much variability, or there's too much heterogeneity, or whatever. And my response to that is, so, like, I mean, if you don't want to deal with that, that's fine. But medicine is all about uncertainty, and we need to be able to sort of grapple with that and be comfortable with it, and try to navigate it. So there are philosophical differences, but it's what's encouraging to me, and why I think that we will figure this out, is that there's just an army of people now who are clinicians and scientists and researchers and advocates, um, and people with lived experience who are willing to grapple with that uncertainty and still, put their time and energy into this, and that gives me hope that we'll figure it
That's
out.
right. Very encouraging. Yeah, very encouraging. And one last question.
Yeah.
Um, and this is a kind of tough one. If you were to look into your crystal ball and say, what are we going to accomplish over the next 5 years? Would you dare to say what you think will happen?
Well, I don't know what's going to happen over the next 5 years, but my hope is that we will identify treatments for different symptomatic clusters, different phenotypes of long COVID,
Okay.
um, that actually reverse this disease and don't just try to mask the symptoms. I think that that is a thing that is possible to achieve, like a cure for long COVID.
Hmm.
Um, it may not be for everyone, but I think that we can achieve that for, I hope, a substantial group of people. But in order to do that, we need to sort of keep our eye on the prize here, and we need sustained investment into all of this infrastructure to do that. And so that means we need the NIH to be fully engaged in this, and funding this work. That means we need the FDA to be supporting this work and providing guidance. It means
Mhm.
we need the patient community to remain engaged and continue to advocate for this. Um, and we need, the scientific community to remain engaged, and not just people who have been working in this for the last 5 years, but we want this and we need this to be a topic area and a field that young investigators want to go into for their career. So
Mhm.
um, I have a lot of hope for the next 5 years based on what I've seen in the last 5 years. And I think that we are getting closer and closer. But we need this sustained investment from like everybody involved in order to achieve that. And so um, hopefully that will keep up. And 5 years from now, we will be reminiscing about our great success in curing long COVID.
Great. Yes, yes, yes. Well, your, the fact that there's so much interest in the field is encouraging, and hopefully, there's so much going on at the NIH. Hopefully, long COVID research will continue to be supported.
It's
Uh,
incredibly important. And so
yeah,
we can't do this without the NIH, and I'm hopeful that their support will continue.
great. Okay. All right.
Thanks
Well,
so
thank
much
you
for
so
having me.
much.
This was really fun. Thanks for letting me just talk so much.
Uh,
Um, but yeah, no, I really appreciate it. And yeah, happy, maybe we can do a follow-up a year from
that would
now.
be great.
Yeah.
That'd be great.
All right.
Great.
Sorry again about the timing snafu, but I'm glad we were able to connect.
No, I'm sure that was me. So,
All right.
all right. Thank you for your patience. Yes.
Yeah, yeah. Thanks so much.
Okay.
Bye.
Thank you. Bye.